welcome everybody to my channel Omni Bioentrance, and today we will be discussing CSIR PYQs of the year 2018. I will be discussing the genetics section. If you are new to my channel, please do like, share and subscribe. So coming to the year June 2018, in an organism, allele for red dye color is dominant over allele for white eye color. A cross is made between a white-eyed male and a red-eyed female. In the progeny, all the males are red-eyed while the females are white-eyed. The reciprocal cross leads to all red-eyed progeny. Based on the above information, which one of the following conclusion is correct? This is a sex-limited trait and the male is a homomorphic sex. This is a sex-linked trait with the male being the homomorphic sex. This is a sex-linked trait with the female being the homomorphic sex. This is the case of autosomal inheritance with incomplete penetrance. If it was autosomal, then reciprocal crosses would have come the same, but this is not the case, so it is a sex-linked trait and the male is homomorphic. This is the condition. This is the male parent X R small r into X capital R Y, that is the female parent. Now cross, you get red male and white female. So when a cross is made between white-eyed male, which is recessive character, that is why I have given small r and capital R for red-eyed female, the progeny, all males are red-eyed. Here the males are red-eyed and the females are white-eyed. So the male is a homomorphic sex. Now this is the reciprocal cross. You will get all red-eyed progeny. So the answer here is this is sex-linked trait with the male being homomorphic sex. Then next one. A male snail head homozygous for dextral allele is crossed with a female homozygous for sinistral alleles. All the F1 individuals showed sinistral phenotype. When F1 progeny were self-fertilized, all individuals of the F2 progeny had dextral coiling. The experiment demonstrated. So this experiment demonstrates not maternal inheritance, it is maternal effect. So maternal effect as the nuclear genotype of the F1 mother has governed the phenotype of the F2 individual. So answer is option 3. And next one. The location of 5 overlapping deletions have been mapped to in Drosophila chromosome as shown below. Horizontal lines in the above figure indicate the deleted regions. Recessive mutations A, B, C, D and E are known to be located within this region. But the order of mutation on the chromosome is not known. When the flies homozygous for the recessive mutation are crossed with flies homozygous for the deletions, the following results are obtained. Letter M represents mutant phenotype and plus represents the wild type phenotype. So we can see that there is a deletion. If you see this column, there is deletion in 1 only and not from 2 to 5. So for 1 we will be putting it as M and the rest will be positive sign. Now we have to see which gene is M for deletion 1 and positive for 2 to 5. That is from the table. So in the table these are the deletions and these are the genes in which mutation has occurred. So it is from deletion 1M to plus, that is seen in the case of B. So it is the B gene which is first. Then if you see this column, it has got 2 deletions. So we put M over there and a positive sign for the regions where there is no deletion. Now if you see for the sequence plus in the table, you will see that it corresponds to C. So the second will be C. Similarly D, E and A. So the order here is B, C, D, E, A. Answer is option 1. The next one. The pedigree given below follows the inheritance pattern of the late onset, that is after age of 30 years. Genetic disease, that is 100% penetrant. Affected individuals are indicated by a solid circle, women, or solid square in the case of male. RFLP analysis of DNA from each individual is shown below in the pedigree. So this is the generation 1, 2 and 3. These are the affected individuals with dark circles and dark square, or shaded circle and shaded square. This is the RFLP analysis. And which grandchildren 3B to 3D will be affected by the disease after attaining the age of 30 years? That is the question. Now if you see the RFLP pattern, you can see that the deceased has two bottom bands two bottom bands. So if it is 1, that is 1A. If you see 1A, it has got two bands at the bottom. Similarly, in the case of 1C, this is 1C, it has got two bands at the bottom. This is 2B. So 2B, if you see, it has got two bands at the bottom. Now which grandchildren from 3B to 3D, so this generation 3B to 3D will be affected by the disease after attaining the age of 30 years. So they should also have two bands at the bottom of the gel. So that can be seen in the case of 3B and 3D. So the answer here is option 4, both 3B and 3D. Answer is option 4. The next one. A chemist synthesizes 3 new chemical compounds in the laboratory and names them as X, Y and Z. After analyzing mutagenesis potential of all these compounds, the genetist observed that all are highly mutagenic. The genetist also tested the potential of mutation induced by these compounds to be reversed by other known mutagens and obtained the following results. So mutations produced by X, Y and Z reversed by nitrous acid, hydroxylamine, acridine, orange. Assuming that X, Y, Z caused any of these 3 types of mutation, transition, transversion or single or base deletion, what conclusion can you make about the nature of mutation produced by these compounds? So nitrous acid and hydroxylamine, they causes transition mutation and they can be reversed by nitrous acid and hydroxylamines. So X we can see from the tabular column, it is reversed by nitrous acid as well as hydroxylamine but not by acridine orange. So X is a transition mutation. Then Y is not reversed by any of them, so it must be a transversion mutation. Z is reversed by acridine orange only. Acridine orange causes single base deletion or frame shift mutations, I can say. So Z causes single base deletion. So the answer here is option 2. X causes transition, Y causes transversion and Z causes single base deletion. Then coming to year December 2018. The pedigree below represents inheritance of an autosomal recessive trait. What is the probability that individual 6 is a heterozygote? This individual is a heterozygote. Now this is recessive, this is autosomal recessive trait, means these parents are heterozygous. That is why you got the 7th offspring as an affected individual. So here it will be 4 will be capital A small a, and 5 also will be capital A small a. So capital A small a into capital A small a, if you cross, you will get 2 by 3 as the answer, because you have to reject this possibility small a, because the individual 6 is not affected with the disease. So small a has to be rejected. And the probability of the individual 6 being a heterozygote is 2 out of 3. So the answer here is option 3, that is 2 out of 3. The next one. The allele L in Drosophila is recessive, sex-linked and lethal when homozygous or hemizygous. If a female of the genotype capital L small l is crossed with a male, what is the ratio of the females and males in the progeny? So X is lethal when it is allele L is lethal when it is in homozygous condition, that is it is lethal for the female if it occurs in homozygous condition, that is XL. It is lethal in the hemizygous condition. So XLY, that is lethal for the male. Now a cross is made between genotype small l, capital L is crossed with a male. What is the ratio of females and males in the progeny? So this is the cross, these are the gametes and these are the offsprings. This one will not survive because it is lethal for the male in the hemizygous condition. But these 3 will survive. So how many females are there? 2 female and 1 male. So the answer here is 2 female, 1 male, option 2. Then next one. Deamination of bases is a common chemical event that produces spontaneous mutation. Which one of the following bases will be formed by deamination of 5-methyl cytosine? So deamination of 5-methyl cytosine lead to the formation of thymine. Answer is option 2. Then next one. Given below is the marker profile for 2 parental lines P1 and P2 and their derived F1 progeny. The marker that is represented in the above figure is most likely to be. So if you see the F1 individual, it is a case of codominant markers, because F1 has got one band from P2 and the other band from P1. So it is the case of codominant markers, as P1 and P2 are the parents and F1 is the progeny. So codominant markers will be RFLP and SSR as per the options. So the answer here is option 1. Then next one. Several mutants 1 to 4 are isolated, all of which require the compound E for growth. The compounds A to D in the biosynthetic pathway to E are known, but their order in the pathway is not known. Each compound is tested for its ability to support growth of each mutant. In the following table, the plus sign indicates growth and minus sign indicates no growth. What is the order of the compound A to E in the pathway? So all plus means, that is over here, plus for E. All plus means that is the end product. That is the end product, and E is required for their growth. So substrate for this E will be 1 plus lesser than this one, lesser than E. So that will be D. Then substrate for D will be 1 plus less than in D. So that will be C. Substrate for C will be 1 plus less than in B. So that will be B. And so substrate for substrate for B will be all minus. So it will be A. So the answer here will be A to B, B to C, C to D and D to E. Answer will be option 4. Next question. Polymorphic DNA sequences are used for molecular identification. Short tandem repeats (STR) and single nucleotide polymorphism (SNPs) are used as polymorphic markers. The table below summarizes the status of autosomal SNP, autosomal STR, mitochondrial SNP, Y-linked STR for 4 individuals related to each other, representing parents and their 2 children. So this is the individual, these are the autosomal markers, this is mitochondrial and this is Y-linked marker. Now markers which are autosomal, they will be inherited from both the parents, while the mitochondrial marker is of maternal origin and Y-linked markers are from the paternal origin. So based on the above data, identify the individual representing the 2 parents. So we can see that A and B are having Y-linked STR marker and they are the males, and C and D are the females. Now based on the above data, identify the individual representing the 2 parents. First one, individual A and D. If individual A and D are the parents, then B and C are their offsprings. So A is having autosomal SNP CC. D is also having CC, but B and C are having CG. So they are not the offsprings of A and D cannot be the 2 parents. So option 1 is not correct. But if you see option 2, individual A and C. A is a male, C is a female. So B and D will be the offsprings. So A is having CC marker and C is having CG marker. Now B is having CG marker inherited from C and D is having CC marker. So SNP marker is as per the parents for offsprings B and D. Now STR 1312 for A and for C it is 1413. So 1313 for B and 1314 for D. That is also correct. Now SNP1 is C for all. SNP2 is mitochondrial SNP2, that is of maternal inheritance. That is G for A and D for A. But A is male parent because he is having the Y-linked STR. So the female parent here is C, A. So A is present in B and D. So that is correct. Now 13, Y-linked STR is present in A. In B also Y-linked STR 13 is present. So individual A and C are the parents and B and D are the offsprings. So option 2 is correct and option 2 are the individual representing the 2 parents. Answer is option 2. Then next one. Using interrupted mating, 4 HFR strains were analyzed for the sequence in which they transmitted a number of different genes to the F- strain. Each HFR strain was found to transmit its genes in a unique order as summarized in the table. Only the first 5 genes were scored. Which of the following correctly represent the gene sequence in the original strain from which the HFR strains were derived as well as the place of integration and polarity of the F plasmid? So the first HFR strain, that is A, B, D, F, H. So we write it over here. The second one B, A, L, M, K. So B, A, L, M, K. Third one M, K, H, J, G, H. So M, K, J, G, H. Fourth one F, H, G, H, G, J, K. So F, H, G, J, K. So if you see all the options, so option 1 if you see A, B, D, F is here, H is here, that is not correct. Now if you see option 2, A, B, D, F, H, then next should be L, M, K, JGH. So LMK JGH. Now we will see for the F factor attachment. So in the first strain, A is the one which is to go first. So we have to put a mark before A, but it is not present in option 2. So that is not correct. Now if you see 3, it is A, B, D, F, H, L, M, K, J, G. So that is correct. Now we will put the marks for F factor. Now A is the first one to go, so a mark before A. So it is over here. Now in the second HFR strain, B is the first one to go, so a mark before B. And in the third HFR strain, M is the first to enter, so a mark before M over here. Then in the fourth condition, F is the first to enter, so a mark before F, so F over here. So option 3 is correct. Answer is option 3.